Hi guys, in this video we're going to talk about practicals and how to devise practicals given different scenarios, perhaps in an exam question situation. Okay, so in my experience it's practical questions that pose students the most kind of issues, and I think it's because not only you have to remember how each practical works, but in many cases the context of the practical is changed in the exam question, or there's just so many things to consider in relation to the practical that it's difficult to know A, what things to put down, and B, what the best structure is to kind of put that all together. So in an attempt to try and address that, what I'm going to do is give you my what I've learned from looking at many types of questions in terms of what are the most essential things that must go into a practical. Now, this one video is not going to be enough to cover everything in relation to practicals, but what I do want to do is at least address the most important things. If we're talking about practicals in general, there are some things that are very important so that if you do an experiment, you can make conclusions based on what you have done. Now, in order to make conclusions on an experiment, which is essentially the most important thing about an experiment, is because of the things that you did, you can make a conclusion from it. Validity is essential, but there are other things to do with practicals as well. So for example, validity is one thing, reproducibility, we're kind of considering the precision, the reliability of what you've done. Was it a one-off, or would it be repeatable? Validity, reproducibility, and then we have potentially accuracy. How well has your experiment allowed you to make conclusions that might be applicable to things beyond the scope of your experiment? So you might have done an experiment on a plant. Is that true for all plants? Is that true for all members of that species? How accurate are your results in terms of what the wider context? And then you have analysis, so data analysis to do with your experiment, the nature of the data, what kind of tests will you put your data through to interrogate how, really what your results are telling you, so that you can make proper conclusions from it. And finally, I would say, we've got risks, risk assessment, health and safety, those kind of things, and even, if we're really stretching it, maybe even ethical concerns to do with what your experiment entails. Does it jeopardize safety to a great extent? Does it involve animals, and so do their rights have to be taken into account? All these things are, you have to consider them in terms of a practical, but what I'm going to focus on today is only the most important one, which is this one, this is validity. And if you get a 3 to 4 mark question on a practical, it's the validity of what you're talking about that's what those 3 or 4 marks are going to be for. You're not going to get 3 to 4, you're not going to get any of those 3 to 4 marks for a practical question where you have to design the whole practical simply for saying, oh, I'm going to repeat my experiment. No, that's unlikely to be the case. Okay, your 3 to 4 marks on making a practical, on designing a practical, is all going to be about how you set that up. Do you remember the method? Have you said clearly what you're measuring? And have you kind of controlled certain things to make sure that your conclusions are valid? Okay, so let's start talking about that. Okay, so what makes a valid investigation? 3 things. It's as simple as this, okay? 3 things, and I'm sure you've heard this in some format or other, but I think even if you know the basics, there are some finer points that should be considered, okay? So first, your independent variable. Your independent variable is the thing you're going to change. Any experiment, something's got to be different okay, or some variable needs to be changed or recorded or observed in order to see or observe the effect of that variable, to see if it's associated with something else. Does it cause a change? Because then you can make a conclusion. So the independent variable is essentially what you change in the investigation. 2, your dependent variable is the thing that you are going to measure. And 3 is the control variables. These are the things that you are going to keep the same. And these are very important. But it is very dangerous to think that simply by stating these that your job is done. It is not. What you have to do, in my opinion, is you've got to talk about not only what they are, so yes, you have to state what they are, good, right? But essentially, I think you're getting the marks for identifying what the independent variable is, and then saying how you are going to change that in a way that addresses the thing that you're investigating or the context of the question. Okay, so again, saying what you're measuring is kind of half the story there. You must do it, but again, what are you measuring and how are you measuring it? And essentially the how in this section is going to be, do you remember the method, and can you tweak the method a little bit to suit the requirements of the question that you've been asked. Finally, you've got the control variables, the things that you're keeping the same. Again, state what they are, yes, but you might want to mention how you are keeping them the same. But again, actually here, I'm going to change this. Here, I don't think you need to go too much into how you're going to keep this the same. It is an important consideration, but let's just call it a bit secondary right now. How is fine. Control variables, I'm going to keep certain things the same. You have to make sure that these control variables are relevant. These are the things that I see most people doing wrong, or many people doing wrong, is they're just picking out some random factors that they're going to keep the same. Whereas really, you've got to think about the factors that are most likely to affect your experiment and choose those as your control variable. Okay? Now, through some examples that I'm going to go through, hopefully this will become a bit more clear. Okay? But essentially, independent variable, say how you're going to change it. Dependent variable, say how you're going to measure it. Control variables, keep them relevant, and it might be enough just to say, I'm going to keep them the same. Okay? Right, let's proceed. We'll look at our examples to see what I mean by this. Let's go. So, the effect of temperature on an enzyme. So that's going to be our first example, pretty basic. Okay, next we'll do an ecological study. We're going to compare the biodiversity, the plant biodiversity. Let's say we're going to compare the plant biodiversity in 2 areas. Okay, so we've got a lab-based enzyme experiment. We've got an ecological study that we'll have to go out into the field to do. And finally, we've got the effect of antibiotics on bacteria. So quite different things. And then we'll see how, regardless of whatever kind of practical it is or investigation it is, these things apply at the core. Remember, there's more to these than just this, but these are the most important. Okay, so if I had a list, and maybe a subsequent video will go into, point number 4 will be how am I addressing how accurate this is. Point number 5 will be how am I addressing how reproducible it is, how reliable it is. Point number 6 would be if there's any health and safety concerns, and how am I addressing those. Okay, let's begin. Temperature on an enzyme, so easy peasy. I'm investigating the effect of temperature on the enzyme, so my independent variable is going to be temperature, and I'm going to change the temperature, but the way I'm going to do that is I'm going to repeat whatever experiment I describe. I'm going to repeat this experiment at a range of different temperatures. I'm going to repeat the experiment at a range of different temperatures. I would always suggest 5 as a very safe number to have a range of temperatures. Remember, if you're doing, look into the question to see if there's any clues about whether the examiners want you to state a particular range of temperatures. But if it's just not stated, then 5 different temperatures would be fine. Okay. Ideally, if you're looking at the effect of temperature on an enzyme, you want to go from 0 to 80, 90. So you want to cover all that range, that from when the enzyme is inactive all the way to when it's denatured. Change the temperature, repeat this at different temperatures. And how would I do it? Well, I would use water baths that have been set up at those different temperatures, at that constant temperature. And my boiling tube with the experiments, with the reaction happening, I'm going to make sure that the enzyme substrates are going to be in that boiling tube, equilibrate to that temperature, and that should be fine. Next, dependent variable. How am I going to measure this thing? Well, again, so this mainly comes down to the revision of your methods or of your core practicals. Have you understood what you were doing when you were doing the core practical? And I would say pay attention during the core practical. It makes your revision so much easier if you paid attention during the core practical, you understood why you were doing everything, rather than taking it a bit lightly because it's a break from your normal routine lessons. Okay, so next thing is how we're going to measure this thing. Now remember, enzymes, obviously, we have boiling tubes, and in the boiling tubes we have our enzyme and we have our substrate, and the enzyme is going to work or catalyze the reaction that's going to convert the substrate into the product. Okay, so we want to add enzyme, so we're going to add enzyme to the substrate that's in the boiling tube, in the boiling tube being in the water baths at a particular temperature. But then the next thing is going to be different for the enzyme that you're looking at. So some enzymes, when they convert the substrate into the product, it produces a gas. So the product might be a gas. Sometimes you might have a color change. So your product might have a color to it that you can measure. In some cases, your substrate might have a color to it, and as your substrate gets broken down by the enzyme, the color starts to go away. So the way usually we can look at whether an enzyme reaction is happening is usually if there's a gaseous product being formed, or if there's some kind of color change that we can measure as the reaction happens. But again, look for the clues in the question. So if it's an unfamiliar enzyme, there might be some information given in the question that will tell you what this enzyme does that could be measured. Usually, it's a gas production, in which case, if it's gas, then we are going to be measuring the volume of gas produced. Okay, measure volume of gas. But if it's a color change, we might be taking out samples at regular intervals and using a colorimeter to measure the intensity of the color to see how far that reaction has proceeded. Okay, right, and then because we're looking at enzymes, it's likely that we're going to be looking at the rate of the enzyme reaction, so it's always useful to mention timed intervals that you are going to be doing these things. But essentially, that's what you're doing. So we've said how we're going to set the experiment up. We've said what we are measuring, we've said how we are going to do it, and that should essentially be enough. But that's not a valid experiment yet, because in order for this to have any meaning, we must make sure that we've addressed our control variables. So if we're looking at the effect of temperature on an enzyme, remember we've got to keep it relevant. Can't just pick out random things like light intensity, because light intensity doesn't affect really enzymes directly. Okay, so that wouldn't have been relevant. So what's relevant to the enzyme? I've got to use the biology. So right, I'm looking at the effect of temperature on an enzyme. What are the most likely factors that are going to affect the enzyme that are not temperature? So I'm thinking pH, I'm thinking concentrations of substrate and enzyme. They are likely to affect it the most. So these are the things that are most likely to be my control variables that I have to keep the same. So pH, I've got to keep that constant. So I'm going to use pH buffers, and I'll use a pH at which I know the enzyme should be active. Concentration, again, you might not have to say too much about this beyond keeping the concentrations the same, but you might also want to say that you want to keep the volumes and the concentration of the enzyme and the substrate the same. You don't want that to factor into how much the enzyme is active. Okay, now that collectively, that is your 4 marks essentially, right? Again, could we say we're going to repeat the experiment? Yeah, but it's less important than these things. Okay, right. Next, comparing the biodiversity in 2 areas. So what is changing in the 2 areas? Obviously there's something different in the 2 areas, otherwise why am I comparing them? So maybe it's light intensity that I'm comparing in the 2 different areas. Maybe one area has been treated with a pesticide and the other one hasn't. So these are the things that are different, and I'm going to choose those 2 areas. So one area with high light intensity, one area with low light intensity. Okay, simple as that. And if it's pesticides, for example, one area that's been treated with the pesticide, and one area that has not been treated with the pesticide. Okay, there we go. So we've got our independent variable, and we have got 2 areas that we're going to compare, and we've said how we are going to investigate the effect of this factor. Then experiment or practical or investigation, like how are we going to look at this? So again, your recollection, your application of the practical methods that you have studied. So in terms of comparing 2 areas, we want to compare 2 large enough areas in order that they are representative for an ecological study. Okay, we want to use quadrats in a random way. Okay, so talk about your random number generators being used to produce coordinates, and place the quadrats at those coordinates so that within each area you're carrying out random sampling. Okay, so large areas to be representative, quadrats, random sampling, putting the quadrats down. But once the quadrat is down, what is it that you're measuring? This is part of your method. What is it that you're measuring? And remember, we're looking at whether the light intensity of the pesticide is affecting the plant biodiversity. So how are we going to do that? Well, once the quadrat is down, we are going to measure or record the abundance of each plant species. Remember, the way you can do that is by looking at the percentage cover, or by using an abundance scale such as the AC4 scale. Okay, so, and it might be useful to mention that you're going to identify the different species using an identification key. All right, so a bit more information goes in this dependent variable section, but I mean, according to me, the method that you have memorized or learned or understood, that needs to go in this section, okay? Right, so we've got two areas, same size, goes here, got two areas. This is what we're going to do in each area. That's going to allow us to measure the biodiversity. So actually, because it says biodiversity, we might want to say, you know, just finish off with saying calculate the biodiversity using the data that we've recorded. So once we've got the abundance, once we've got the different species, we should be able to calculate biodiversity. Okay, and then compare. Now, in order to make that comparison fair or valid, what were the things that we could have controlled? And remember, for an ecological study, you can't really control anything. You can't control the pH, you can't control the temperature, you can't control the weather, anything like that. So what you can do is do certain things to ensure that the comparison is valid. So what you can do is make sure that the areas are the same size. The areas are the same size. You can make sure that the method that you use in the two places was consistent. And this is all really about the same. Okay, so same method, same size. What else could we do? We can make sure that the areas were sampled at the same time of the day or the year to make sure that none of those other factors could be affecting the results that we get. And yeah, that's all I can think of at this time, but that's the kind of thing that you're looking for there. Next, we've got the effect of antibiotics on bacteria. The independent variable in this case is going to be a range of different antibiotics. Okay, we want to test the effectiveness of those antibiotics, so we're going to have a range of different antibiotics, and I think it might be enough to leave it at that. Okay, in fact, we might, well, that's kind of in the control variables thing. Okay, fine. So different antibiotics, and how are we going to measure their effect? So what we're going to do is we are going to inoculate agar plates with bacteria. We're going to inoculate some plates with bacteria, and we're going to spread them out to eventually ensure that they grow as a lawn, an even layer of bacteria over the plate, over the agar. Okay, and then once we have done that, we will place discs soaked with the antibiotics. Okay, so place the antibiotic soaked discs on the bacteria, on the agar, and then we're going to incubate the plate for a sufficient amount of time to allow the growth of the bacteria, 24 hours, 48 hours. Okay, and then depending on the effectiveness of the antibiotic, there's going to be a different size of clear zone around that disc. So we are going to measure the diameter of all the clear zones. Great, easy peasy. Right, so we've got different antibiotics. We're going to expose the bacteria to the antibiotics, allow the bacteria to grow, measure the clear zone, and that's fine. However, it's not valid until we make sure that certain things are controlled. So, we want to make sure, all right, so again, we've got to keep it relevant. pH, temperature, not really critical factors here. In order to be able to compare the different antibiotics, the most important thing would be to make sure that actually the bacterial population is genetically pure. So, you know, you don't have different types of bacteria in there. It's not a mix. So you maybe have one type of bacteria maybe growing at that end of the plate, another type of bacteria growing at this end of the plate, and therefore the different sized clear zones is not really because of the different antibiotics. It's more to do with that there's different bacteria growing at different ends of the plate. You don't want that. Okay, so in order to make sure that the comparison is valid, we must make sure that the bacteria is a single population of, or at least they're all the same. Okay. Next, we want to probably make sure that the size of the discs that we use are the same, because we don't want the amount of the antibiotic to be the factor that's affecting how effective it is. It's the antibiotic, the nature of the antibiotic itself that we're investigating, not how much of it there is. And maybe concentration of antibiotic. We probably want to make sure that the concentration of the antibiotic that we're using to soak the discs, that's the same. Okay? And if we keep these things the same, then the different size clear zones that we measure here, they're a valid comparison. Okay? So that's where I'll leave it. So I think for most exam questions to do with practicals, this is what you need to focus your attention on. Okay? If your practical starts to go 5, 6, 7, 8 marks and beyond, then we need to start talking about repeats. Then we need to start talking about accuracy. We need to start talking about health and safety, risk assessment, ethical considerations, and so on. But 1, 2, and 3, these factors are important. Don't just identify them. Say what you're going to do to change them, and remember, think about yourself, think of yourself doing the experiment. Don't just think of it as some facts that you're slapping on the page. Okay? See yourself doing these things. That will give your answer the best structure once you've given all these things consideration. All right, guys, good luck.